So what happens next? Well, this would be oxidative phosphorylation. This is what happens. But before we get into that, let's review our totals for 1-glucose. From glycolysis, we made 2 ATP, and that is net. We formed 2 NADHs because we picked up electrons and hydrogens from our sugar molecule, and in the end we formed 2 pyruvate molecules. From there, we oxidize our pyruvate, and we form, these are totals for both pyruvates. So we get 2 CO2s, and we get 2 NADHs and 2 acetyl-CoAs. And lastly, the Krebs cycle. Our 2 acetyl-CoAs are then further oxidized into 6 NADHs, 2 FADH2s, 2 ATPs, and 4 CO2s. The CO2s are going to be waste products, ATPs are used for energy, and our electron carriers, NADH and FADH2, are going to go and be oxidized. Remember, they are in their reduced forms. So in the inner membrane of the cristae of the mitochondria is where oxidative phosphorylation takes place. The big green proteins are protein complexes that are also hydrogen ion pumps. And at each of these, NADH comes in and drops off its 2 electrons and hydrogen ions and is oxidized back into NAD+. This NAD+ will go back and pick up more electrons from the Krebs cycle, oxidation pyruvate, or glycolysis. FADH2 comes in at a slightly lower energy. It drops off its 2 electrons and 2 protons and is oxidized back into FAD. This is where the oxidative part of oxidative phosphorylation comes into play because our electron carriers are being oxidized. And as those electrons are passed from protein complex to protein complex, we're using their energy to move hydrogen ions into the intermembrane space. And those electrons are being pulled toward oxygen because of its electronegativity. When it does that, oxygen gas molecules are split. Each atom picks up 2 electrons and 2 protons to form water. The phosphorylation part of this comes from ATP synthase that uses the hydrogen ion gradient to form ATP. We're going to look at that in a minute. So let's recap oxidative phosphorylation. Electron carriers are oxidized at the protein complexes. Electron carriers are oxidized at the protein complexes. Then those electrons are going to be passed down. As they move down, they lose energy, pumping those hydrogen ions into the gradient. Electrons are going to flow toward oxygen because of its high electronegativity, where it is reduced. When oxygen is reduced, we form water. And then lastly, the proton gradient will be used to power ATP synthase to form ATP through proton motive force. So here's ATP synthase. Hydrogen ions are going to diffuse. It is like a rotor, and when hydrogen ions diffuse, it turns the rotor, giving motion. This motion is going to drive the catalytic component of ATP synthesis. Remember, that's a highly endergonic reaction. We need to generate a lot of energy to join that inorganic phosphate to ADP. So the motion of the hydrogen ions diffusing down its gradient is going to turn the rotor, which causes the proton motive force, which causes the synthesis of ATP at the active site. So the whole point of the electron transport chain is to use the energy of those electrons to build the gradient that allows ATP synthase to work. We find electron transport chains not just in mitochondria, we find them in bacteria, we find them in chloroplasts as well. Very similar structures, very similar processes.